Everyone's here. Welcome to the 3-minute thesis, or 3MT for short, as hopefully all you know that. My name is Chandra Zenner Ford, and we're starting on time, thankfully. I'm the CEO of Southwest Idaho for the University of Idaho, your host school for this event, and it's the 4th annual. So I was able to do it virtually last year, so this is my second time. I had a great time seeing all the presentations, and I very much look forward to things today. This celebration of student research and the results of their focus on creating new knowledge reminds us all of the impact of higher education in Idaho. We want to welcome all the people on Zoom, students, family, friends, parents, faculty, everyone listening from BSU, University of Idaho, and ISU. Welcome, and we're glad you were able to join us via Zoom. We've got a room full here participating too. The winners of today's competition will be able to compete at the Western Regional Competition in March. So there's a next step after this competition. And one thing I want to remind those of you on Zoom, please make sure you're muted so we don't have any background noise when these students are doing their presentation. So just a reminder of that. And then a few other things we're going to do before we get started. We have a few features from the leaders of the institutions. We have some thank yous to give out and everything. So we've got a few minutes here before we get rolling with the students. And in the spirit of shining a light on the contribution of the 3 participating institutions of higher education in our state, we're going to feature a message from all 3 presidents, Marlene Trump, Scott Green, and Kevin Satterley. So we'll see those in a bit. And they're going to do a little bragging about the Broncos and the Vandals and the Bengals. So you can enjoy that a little bit later. A little bit of housekeeping as well. The QR code feature on your phones, and scan the link so that you can scan the link on your screen. This will give you detailed information on your student presenters and their representatives from each of their institutions. The link to the digital program will also be available on our U of I's website on the 3MT page, and it's going to allow you to vote at the end, and Jerry McMurtry will tell you a little bit about that voting, so everyone here gets a chance to weigh in. Also, for our online audience, please make sure your Zoom screen is set up to side-by-side speaker view for the best possible viewing. You will see the view option noted in the upper right corner of your screen. Choose the side-by-side speaker option to enjoy the show. So now, before we move on, early on when we set this date, we weren't really sure as the host school that President Scott Green's schedule would allow him to be here in Boise. But lucky for us, President Green is here and in person, and he's going to be here to listen to all the pitches from the students across the state. As many of you already know, President Green has made research a top priority for the University of Idaho and dedicated investments to incentivize Idaho's research enterprise. You will see his formal remarks in the video clips later that all 3 presidents provided. But I just wanted him to come up for a few minutes and give a special welcome since he was here in person. So, President Green?
Yeah, you're going to hear from me a little bit later, so I'll try not to bore you with too much here, but I just wanted to spend a short time here to welcome you to the 3-minute thesis Idaho statewide competition. I mean, this is amazing, and I know how excited you all are. I can't wait to see your presentations. This is really kind of one of the highlights of my year. As president of the University of Idaho, I take great pride in the fact that we host an event which shines a light on research in Idaho. And here today, Boise State, Idaho State, and U of I students share some of the emerging research projects and ideas which will help us discover more about the world around us. At the University of Idaho, our students, faculty, and staff and alumni contribute directly and indirectly to more than $1 billion to Idaho's economy, and all of the research institutions and the education institutions in the state, including our 2-year schools, generate more than $4 billion in economic activity. Pretty amazing, right? Research really drives innovation, economic development, and discovery, and Idaho's 3 largest institutions of higher education provide the infrastructure and the resources to enable our students and Idaho industry to thrive. So I want to thank Jerry McMurtry. He's the dean of our graduate school, for being the leader and producer of this event. It is no small undertaking to put all this together and get everybody here. Finally, I also especially want to thank all the students for participating in the effort that you put into preparing your presentations for today. We will all benefit from learning about your work. I'm deeply grateful for that, and I'm really looking forward to it. So thank you, and good luck.
Thank you, President Green. I'm super proud to be a part of his team and excited to be here. President Green, Kevin Satterley, and Marlene Trump we're in a new era of cooperation. So, and I know today we're competing, but it's a friendly competition. So this is a good place to be. So I want to take a moment to thank our judges and introduce them. This group of 5 is an amazing group we have this year, and I'm super excited they all accepted our invitation. So I'm going to start over here with our veteran judge. He is in his second year judging, and last year we did it all virtual. And Matt Weaver, who is the Deputy Director of the Idaho Department of Water Resources. As I said, he graciously volunteered last year. He has a civil engineering degree from Montana State and a master's of earth science from Boise State. And his office just so happens to be up above ours, and we are happy to partner with them in the Idaho Water Center. Thank you, Matt. Next is Nick Miller, who happens to be the executive director of the Venture College at Boise State, but he's also a proud Vandal graduate. So I remind him of that with all his work at Boise State. And he is on the Boise Advisors group as well, and I want to thank you, Nick, for being here. Let me see. Next is Representative Lori McCann. She represents District 6, is from Lewiston, and has an undergraduate degree from the University of Idaho in education, and she has served as a professor at LCSC. And this isn't, she doesn't live in Boise, but she's down here for the session, and so we were glad to snap some of her time and have her come over here. So thank you, Representative McCann. Next is Callie Roach, and Callie is the newest member of the Idaho State Board of Education. She is the retired vice president of Clear Springs Foods. Did I get that right? I always want to say Clearwater, out of the Magic Valley. And Callie earned an undergraduate degree from LCSC and a master's degree from Idaho State University. Thank you, Callie, for being here. And then last but not least, we have T.M. Rastegar. And T.M. is the executive director of Trailhead and has graciously partnered with us to support this event and use this space. And I think there's a lot of alignment given that this is the space where entrepreneurs launch their business. And some of these ideas today just might be the next entrepreneur of tomorrow. So I'm happy to be here, Tim. And you earned your undergraduate and MBA from Boise State. And I made the point of sharing everyone's educational backgrounds to show that we covered everybody. There's no bias here. We're all in it for all the right reasons. So thank you for sharing your time and talent with us. And then I have a few other thank yous to make, and that would be the deans of the graduate schools from all 3 institutions. Scott Lowe of Boise State University, Adam Bradford of Idaho State University, and of course Jerry McMurtry from the University of Idaho. And then you'll notice on each slide that the students shared their major professor. And I think we all should take note of that because every one of these faculty played a role in the student being here today, and we should all be grateful for the time they've spent with the students and getting them here. So a special thank you to all the faculty. Hopefully some of them are here on Zoom. And then last but not least, this does not happen without a lot of details and logistics. And I want to give a shout out to the University of Idaho staff who collaborate and pull together all the details for 3MT. From our Boise team, Donna Stolaroff, Maria Ortega, Troy Jacoby, and Teresa Amos, and from Moscow, Lauren Perkinson and Stephanie Thomas. We so appreciate everything you've done. Thank you, thank you, thank you. All right, we're almost ready for the competition. But first, we're going to hear from all 3 leaders, President Marlene Trump, Boise State University, President Kevin Satterley from Idaho State University, and President Scott Green from the University of Idaho. Please watch your screens for some encouragement and inspiration from our 3 fearless leaders.
Graduate education is so incredibly important, and not just for your future, but for the future of the whole world. It's so exciting to be a part of this event because universities don't just share knowledge, we create it. And each of you are knowledge creators, impacting the world with your new insights. You are forever a part of a university that is committed to your continued success as students and as professionals, and one that is positioned to be a difference maker for Idaho and far beyond. This event is a true highlight of the year, and I'm so excited to hear about all of the incredible work that your time and passion have created. Congratulations on being here, and I can't wait to learn more about your projects.
I want to welcome you all to the statewide 3MT competition. I want to begin by saying congratulations to all of you who have spent countless hours conducting the research that you are presenting here today. Each of you represents the value and importance of how graduate education is significant for bringing change to our society. Conducting research as a student is an important part of the modern university learning process. That is why at Idaho State University we recognize the importance of our students pursuing new knowledge to better our world. Because so much of learning is not just listening, reading, or passing information from one another. Much of learning is the experiential factor of helping to bring about those new discoveries. By doing so, you are helping our society pursue new knowledge through research. As some experts put it, the pursuit of knowledge and discovery has always been an intrinsic human characteristic. But when new knowledge is curated and put in the right hands, it has the power to bring about high-value change to our society. And now you can demonstrate your skill by condensing all of that into a 3-minute expression of your concept. Because being able to summarize months of your research into a 3-minute discussion is an important life and career skill. So I'm glad you're doing it today. I want to congratulate you all on what you are doing and all that you have done to get here. I also want to say thank you. Thank you for taking such an interest in your education and making the most of your future. Good luck, and thanks for participating here today.
Today we will watch our best and brightest graduate students discuss the research they've poured thousands of hours into. They will articulate why this research matters, and their passion for their topics will shine through. The quality of their work demonstrates the strength of our research programs in Idaho. We'll send today's winners to regional and national 3MT competitions. The topics today vary from science to the humanities and showcase the results of investment in Idaho graduate students. Our state, the nation, and our world are improved by their research. The U of I and our sister institutions are shaping citizens who contribute to Idaho, a bright group of doers and thinkers who care about the planet and will propel our state's economy forward. The U of I invests more than $100 million on research activities each year, from sending antibacterial polymers to the space station to building a rotorcraft that will go to Saturn's moon, Titan. From developing new drought-resistant grains to studying vector-borne disease, our research advances our society in impactful ways. The work presented today will benefit not only our communities, but will also help us address some of the world's greatest challenges. Here in Idaho, we are creating teachers, doctors, nurses, scientists, and farmers that will go on to make a huge impact on our state and our country. We are committed to supporting transformative student experiences. At the University of Idaho, 66% of our undergraduates participate in hands-on research, and we award over 600 teaching and research assistant positions for students each year. We are proud of the life-changing education we provide at the University of Idaho, Boise State University, and Idaho State University. We will continue to deliver accessible, high-quality education at a great value for the state of Idaho. We have 3 great institutions in Idaho working on research that impacts lives around the world. The 3MT event demonstrates how we can take complex, detailed academic research and make it understandable to the citizens of our state. Most importantly, this event showcases how our research will impact our communities and the world. Enjoy the 3MT competition.
All right. So before I have Jerry McMurtry come up, I'm just going to say one quick thing too. He's going to go over some housekeeping and rules and guidelines for everyone. When you're done with your pitch, just stay on the stage for a minute because I'm going to ask maybe a question or two to just have some time in between each presentation for the judges to take notes. So don't rush off. Just let me ask you a few questions before you're done. So Jerry, you want to come up and do the guidelines?
Thank you, Chandra. You guys are the best of the best, so you already know the rules, right? Okay, so in the back you'll see a timer, and you're familiar with 3 minutes, 180 seconds, and so you've got that back there for you. As you know, your presentation can't exceed 3 minutes. Please stay on stage with Chandra. She will have some fun questions, and you can take your masks off while you present, and so that's absolutely appropriate. When you are done presenting, you'll have a handheld microphone. If you could just hand that to Donna here at the side of the stage, she'll go ahead and sanitize the next microphone and hand it to the next competitor coming up. For those of you in the virtual audience, please put your questions in the chat, and we will be able to answer those or ask those questions of the competitors at the end of the event. And please don't forget to make notes about your favorite competitors, because you will have a chance to vote for the People's Choice. And so at the end of the event, at the end of all of the presentations, you'll see a slide come up that'll have a QR code and a link, and that's where you will access the ballot for the People's Choice. So with that, Chandra?
Thank you. Now we get to the best part. So I hope our first presenter is ready, because it's time for Rebecca Miller from Boise State University to join us on the stage.
Imagine the next time you go on your weekly grocery store run, trying to purchase everything that you need without getting anything packaged in plastic. That would be hard to do, because plastic packaging has become essential for the protection and preservation of consumer goods. Now, unfortunately, over 56% of all plastic packaging produced is not recyclable due to material complexity. An example of this is multi-layer flexible packaging. Some images of which you may be familiar with of these types of materials are shown on the left-hand side of my slide. These multi-layer systems are useful because they have versatile properties and enable an increase in the longevity of shelf life of many products. However, there currently is not a feasible separation system in place for the various layers, which is why it is not recyclable. You can think of these materials being similar to cooking an egg on a pan without any oil. The egg cooks fine, but there's no simple, easy, clean way to remove the egg from the pan. Without a feasible separation system in place, these materials end up in our landfills and environments, taking hundreds of years to break down. In fact, every year there's over 8 million metric tons, or approximately 100 cruise ships worth of plastic packaging alone that gets discarded as unrecyclable waste. To combat this, my research focuses on making these materials recyclable through enabling a separation system via degradable polymers. Specifically, I design new types of polymers that can come apart or degrade on command when exposed to a specific stimulus. Integrating these polymers at the various interfaces of the different layers within multi-layer packaging can facilitate their separation. Likewise, I've made a design that's like adding oil to the pan when you're cooking an egg. The egg still cooks fine, but is now easily removed when desired. Now, I can tune these polymers to come apart in several different environments, but I've been able to selectively tune them to come apart in environments and processes already used in industry for washing and recycling plastics, meaning that this is easily accessible and applicable on a large scale right away. This may seem too good to be true, but it's not. We know that this works. We've demonstrated with several proof of concept experiments that layer separation can successfully happen when triggering the degradable polymers to come apart, allowing the various layers to be collected for recycling. This work is filed in a provisional patent, and we are excited to have it pave the way for large-scale recycling of complex plastic packaging materials. With my work, we can continue to reap the benefits that plastic packaging offers while limiting the amount of plastic waste that we contribute to. Thank you.
Good job being the first. That's hard. Good job. So I'd like to know what are some of the stimuli that make it all disappear?
Yeah, that's a great question. Some common ones are UV light, heat, or pH. And pH is actually what we use for making it compatible with industry.
Okay.
So, yeah.
So what got you interested in this? What inspired you to do this research?
Well, I was first interested in Boise State, didn't know a lot about what my actual, specific project would be, but frankly, just having a constant motivation like the plastic waste issue is really important, I think, for keeping me driven throughout this process.
Great. Well, thank you. We appreciate your presentation, and good job.
Great,
Yes.
thank you.
Good luck. Thank you.
Thanks.
All right, thank you, Rebecca. So our next presenter is Allison Loyola from Idaho State University.
When you leave tonight, the only thing you will remember is that your brain is like a garden. It is the home to several unique cells. We will focus on two, microglia and neurons. Both of these are particularly important in the development of the brain. Let's imagine the microglia as the gardener and the neurons like the plants. In a healthy brain, the gardener does an extraordinary job during development, pruning the neurons, allowing proper signals throughout the brain to carry its functions correctly. What would happen if the gardener becomes dysfunctional? The plants become bushy and unhealthy. In the brain, we call this hyperconnectivity. Did you know that hyperconnectivity is one of the hallmarks of autism? 1 in 44 kids in the US is affected by this condition, and there is no cure yet, despite such a high incidence. So, what would make microglia dysfunctional? One possibility is drugs. Valproic acid, or VPA, is a widely used drug to treat epilepsy, but taken during pregnancy can lead to an increased risk of autism in children exposed to it in prenatal life. In my thesis, I am working with some celebrities. They are mouse models. These little animals assist us in studying the impact of VPA on microglia. The brain on microglia. Oh my gosh. In these pictures, we can see on picture C, we color-coded the brain microglia with the green dye and the nucleus with the red dye, and photographed them under a very special laser microscope. In picture C, you can see in the lucky mice that did not get VPA, we see healthy microglia with the green and the merge of the green and the red. In picture D, in the not so lucky mice that got VPA, we do not see microglia. When we quantify it, we see a dramatic reduction of microglia in the bottom panel. This is exciting because this is the first clue that tells us the drug is reducing the microglia number, making them dysfunctional at crucial developmental time points. We have a long way to go to decode this medical mystery, but we're enthusiastic that our findings will have pathological implications and may lead to the discovery of new drugs to treat autism. But as they say, one step at a time.
Thank you. I was thinking the whole time, how do you explain this in 3 minutes? But a very complex subject. But what is your major, for starters?
Pharmaceutical sciences.
Okay. And did you have a special interest in autism?
Yes, so I actually have a couple cousins that are diagnosed with autism, and then when I met my professor and she was interested in doing this research, I didn't hesitate and I didn't doubt it. I just wanted to learn more and research and find therapy and a solution to give my cousins or kids like them a better quality of life.
We wish you lots of luck. Thank you. And good luck.
you.
Thank you. Okay, Allison, now thank you for your presentation. All right, number 3 is Morgan Stegmiller from the University of Idaho.
People have many different characteristics, different eye colors, different hair colors, different heights. These are all a result of the genetic diversity encoded for in our DNA. Similarly, sheep have many different characteristics, such as meat, fiber, and wool. There are many different breeds of sheep, such as the Targhee, which is a wool breed actually created right here in Idaho. There's also the St. Croix, which, as you can see from its long flowing mane, it's a hair sheep, and it's known for its parasite resistance. There's also breeds that are known for their fertility, such as the Romanoff. Using all of these different characteristics, many of them are encoded for by single nucleotide polymorphisms, or SNPs, and these are just a change in a single base pair at a location in the DNA. We compared 2 different programs to search through the 2.7 billion base pairs that are in the sheep genome in order to identify those SNPs associated with biological changes. Freebase, which is in purple, identified more SNPs than GATK haplotype caller in all of the breeds. We can then use this data to look at the differences and similarities between the breeds. The plot on the right clusters these breeds by genetic similarity. Each dot is a single sheep, and they are color-coded by breed. As you can see from this plot, there's a lot of genetic diversity between these breeds. We have the wool breeds, like the Targhee and the Rambouillet, up in the top left corner, and then all the way to the right of the plot is the St. Croix, that hair breed. Down in the bottom of the plot, the Romanoff pulls out. We can use all of this genetic diversity in order to understand the genetic causes for economically important traits, such as fiber, meat, wool, parasite resistance. Knowing all of this, we can also show the biological processes involved, not just in sheep, but that can be used as a model organism for cattle and even humans. We also know that over 65% of sheep in the world are located in developing countries, and this makes them vital in providing food for a growing population around the world. We can use the genetic knowledge that we have here in order to understand the biological processes behind many of these traits. From this, we hope to use this knowledge to identify and improve this genetic variation so that we can improve the U.S. industry in sheep, as well as provide food and knowledge of these issues for different things like infertility around the world.
That was great. So are you in the College of Ag?
I am in the
Life
College
Science?
of Ag,
Okay.
yep.
Okay. Your dean is here watching. Good job. I noticed he's here. So what is your major?
Animal science.
Okay, that's what I thought. Did you have sheep in 4-H and do stuff like that when you were growing up? Or?
I actually had alpacas in 4-H.
Oh, okay. I kind of guessed you might have been. But so
yeah.
what got you interested in this research?
Yeah, so I've been interested in animal genetics for a long time, and I'm actually from Ohio, and coming out to the University of Idaho, my major professor was like, look, we have some sheep projects, and I was like, that's really interesting, getting to look at all this genetic variation and hopefully, improve the industry, which is, a great thing to do.
Great, great. Well, thank you and good luck.
Thank you.
All right, our next presenter is Jacob Manzi from Boise State University.
Our electronics are constantly improving, changing, and sometimes in need of repair. What do you do when your electronics break down, when your phone needs a new antenna, battery, or screen? You probably take it to the Geek Squad or the Apple Genius Bar, right? Or maybe the more adventurous and tech-savvy among us take a trip to Radio Shack or order the parts you need to do a DIY repair or upgrade. Now, put on your space helmets and picture us as a civilization living 50 years in the future, on the moon, Mars, or somewhere else. There's no Best Buy or Radio Shack to make sure your iPhone or TV remote are in working order. Plus, it costs about $10,000 per pound sent into space, so I don't think Amazon Prime will ship to you either. My research is in developing a novel technology called plasma jet printing. Uniquely, this printer uses a small jet of plasma to spray nanomaterials onto a substrate and make what we call a printed electronic. In the figure on the left, you can see the black flecks are the nanomaterial being passed down through the purple plasma and onto the flexible yellow substrate below. The inset shows one version of the printer that we built on campus that you can clearly see the purple plasma beam coming out to the side. Now, there are other printers that can print materials, such as inkjet or aerosol jet printing, but they all require a post-processing step, usually involving high heat or intense UV light. These steps are costly, but crucial for the functionality of the printed devices. They also limit what substrates can be printed on, as they need to be able to withstand this high heat treatment. Now, because of the plasma in my setup, there's no need for post-processing, as the materials come out of the printer fully functional and ready to use. The plasma also doesn't burn or melt the substrate, so even paper and thin plastic can be printed on where they couldn't before. The plasma also makes it so that printing is independent of gravity, and a group recently showed on a zero-G flight that the plasma jet printer works in space and microgravity environments and can even print in any direction. Now, during my time at Boise State, I've printed parts for flexible thermoelectric generators, a device that converts thermal energy into electricity. Think of it as something that might charge your Apple Watch using the heat of your body. I've also printed flexible and conductive silver and gold wires, and I'm working toward printing a functional antenna, as well as strain and optical sensing devices. This research is contributing to the ability to be able to print multiple materials onto one flexible and conductive substrate, like the one the astronaut is holding in this image on the right. Thankfully, this technology won't be for a civilization 50 years in the future, but functional in the next several years for both astronauts and civilians to be able to print flexible batteries, solar cells, biosensors for health and fitness monitoring, and more. Thank you.
Thank you, Jacob. That's like science fiction a little bit when I hear it. So you talk about printing batteries. I sort of have a hard time getting my arms around that, although, given all the waste we have with batteries that we use up and everything. So how does that work?
Yeah, so essentially you could find all of the materials that make up a battery and be able to print them instead of like encased and like rolled up inside of like a D-cell battery that we have, but be able to print them on like a flat substrate or do the same thing as long as you can get the materials into ink form.
The heat form. What's your major?
I'm an electrical computer engineering major.
Okay, and where are you from originally?
I'm from Monmouth, Oregon, just outside of Salem.
Okay, well, thank you. Good job.
Yeah, thank you very much.
I have another video for you when we take a little pause. I think the volcano guy was our People's Choice winner last year, if I'm remembering right. Yes, shaking your head, one of those volcano people. All right, so moving on. We next have Arina Ranjit from Idaho State University.
How many of you have drugs in your home? Not for views, but for medical use. Much like me and everyone else in this room, we all have purchased drugs in some way or the other. But did you know that it took 12 years and $4.6 billion for only one drug to have made it to your home, despite other 5,000 similar drugs were tested in the laboratory settings? Many drugs, like a novocaine, that are effective for rheumatoid arthritis, they are not in a drug market just because they suffer from short half-life, which means they are easily degraded by the enzymes of the body, rendering it unstable, giving it no time to stay in the body and show their effects. So the point in a drug research is just not about the discovery, but also about maintaining its stability. That's where my research focuses on, where I conjugate drugs like a novocaine to the long linker, which protect it from the enzyme and further connect it to the moiety that would target the tissue, collectively called novocaine conjugate. When administered, moiety get attached to the bone, act as a reservoir, and linker protect it from degradation, but over the time, drugs get released. That's how it maintains its stability. To prove my hypothesis, I tested in the rats where I induced them with the rheumatoid arthritis. Poor rats though, but for science. So I divided rats into the different group where they received no treatment, another group received novocaine, and the final group received novocaine conjugate. After a few days of the treatment, I saw there was substantial reduction in the arthritis level in a rat treated with the novocaine conjugate groups. This means just by extending the half-life, I could change how drug would work in the body and improve its efficiency. Rheumatoid arthritis is just the beginning, as it could be stepping stone for all those potential drugs which can be cure for a huge range of disease we have in our mankind, as it could pave a way for those drugs to come in a market and make it to your home. Moreover, it would also prompt to use our drug research more innovatively, rationally, economically, and most important, timely. Thank you.
Great job, Arina. So I'd like to ask where you're from.
I'm from Nepal.
Okay, what brought you to Idaho State?
Sorry, what?
What brought you to Idaho State University?
So I'm interested in the drug research, and that brought me here in ISU.
Okay, and what spurred that interest in the research? What got you interested in drug research?
So we all know that whatever progress we have made in uplifting quality of life is largely attributed to the advancement in a drug. And we see how small drugs are and how big their magic is. So I want to be part of that magic and be that someone magician.
Great. Thank you. Good job. Thank you. Good luck. All right, next we have Amanda Bauer from the University of Idaho.
Picture yourself in your kitchen after a long, hard day at work. You're starved. You heat up some leftovers and decide to also make a salad. So you pull out the lettuce and consider washing it, but shrug your shoulders and think, what's the worst that could happen if I don't? We've all been there, not washing our vegetables, but what if I told you that on those leaves are little bacteria that if you ate them could make your medicine less effective? In my research, I study a farming practice that could make this scenario a reality. This practice is the conversion of arid and semi-arid lands, known as dry lands, into agriculture fields. This is happening all over the globe. In fact, 25% of global drylands are currently being used as farmland today, supporting local food security and economies. Indeed, this has happened in southern Idaho. In the picture on the left is what southern Idaho used to look like, very dry and barren. And the picture on the right is what southern Idaho looks like now, very moist and lush after humans have irrigated it. But just like with any other human-induced change, there can be some unintended consequences. In this scenario, by irrigating, we've completely changed the conditions in the soil for the organisms that live in the soil. These organisms include bacteria and fungi, collectively known as microbes. Even though you can't see them, these microbes are very important because they cycle the nutrients plants need to grow. Scientists know that in very lush, moist environments, there are a whole host of bacteria in the soil that are resistant to antibiotics. But in more dry, barren environments, there are less of these resistant bacteria. What we don't know is what happens when we go from one to the other, like what's happened in southern Idaho. For my thesis project, I look at this unknown, asking how does increased moisture from irrigation impact the abundance of antibiotic-resistant genes in the soil? We ask these questions because antibiotic resistance can transfer from the soil to the crop via the bacteria living on the leaf surfaces. This potentially exposes us to the antibiotic resistance when we, say, eat that unwashed salad you had for dinner. This is concerning because antibiotic resistance can make medical cures less effective when we have infections in our bodies. Thus, with this research, we hope to have a better understanding of how desert farming can impact the soil bacteria and the implications to human health. Thank you.
Thank you, Amanda. I grew up on a dryland farming where we didn't irrigate, so I'm curious to know if you grew up in southern Idaho where all this is happening.
I did not. I grew up in California, where we irrigate a ton.
Okay.
Yeah. And so this was a really fun project because I get to look at how irrigation, which I'm used to in California, can affect the soil.
Right. And so are you also in the College of Ag and Life Sciences?
I am.
Yeah, what is your major?
My major is in soil and land resources.
Great. All right. Well, thank you.
Thank you.
I know I wasn't as smart as any of these people when I was an undergraduate or graduate student. Very impressive so far. All right, our next presenter is Jessica Bernardin from Boise State University.
Microbes are bacteria and fungi that are virtually invisible. And despite their small size, they're found not only on, but also in all living things. The natural world is teeming with microbes. In fact, the average person carries around between 2 and 6 pounds of microbes with them every day. Don't worry, the majority of those microbes live in harmony with their hosts and play a vital role in human and ecosystem health. And even though microbes are very small, they form complex relationships in the communities they live in. Now, a microbial community isn't too different from a human community. If you think of your favorite ski town as a community, each resident does a different job. The barista, gear rental guy, food service, but together as a whole, the community's job or function is a ski town. This is the same for microbes. Groups of microbes can do a job together, and that job can change depending on the resources or the environment. Now remember, microbes are just one member of even larger communities. Like the lake pictured here, the microbes are interacting with the plants and the animals and the insects, with each member doing an important job. My research works to understand the types of jobs microbial communities perform and how those jobs might change. But doing a large-scale experiment on a 1,000-acre lake is tough. I have to be able to measure and manipulate the microbial community. So in essence, I need a miniature version of an ecosystem, and that's where pitcher plants come in. Each leaf on a pitcher plant is a cup, and it captures the rainwater and hosts a diversity of organisms, from insects, arachnids, and microbes. Imagine a whole ecosystem that you can hold right in your hand. I use pitcher plants and the microbial communities associated with their leaves to test hypotheses about microbial community function. But why do we care about the teeny tiny microbes on a random carnivorous plant? Well, I studied the pitcher plant system, but you too are a standalone ecosystem. And the microbes that live in you are directly related to your health, just like the microbes on the pitcher plant are related to its health, and how the microbes in that lake are related to the health of the entire aquatic ecosystem. I'm interested in understanding how communities respond to a changing world. From fire and drought, flood and storms, changes becoming more frequent and more extreme. My research works to understand how communities respond to these changing environmental conditions with the hope that understanding these processes on the small scale can help us to protect our planet's diversity on the large scale. Thank you.
So, Jessica, what is your major? What got you down this path?
I'm in the Ecology, Evolution, and Behavior PhD program at Boise State, but I've always been interested in plants and microbes and the fact that there's so much diversity right on surfaces that we don't even know about.
Right. So how far are you from earning your PhD?
I am in my second year, so 2 years to go.
2 years to go.
Yep.
Great, and thank you.
Thank
you.
you.
Did a great job. Thank you.
Thank you.
Okay, our next student presenter is from Idaho State University, and it's Christoph Kissoon.
On April 13th, 2021, Marcus Anthony Singh, a 27-year-old Guyanese-born immigrant to Trinidad and Tobago, was found murdered in his apartment. Clad only in his underwear, Marcus was discovered on the floor, face down, stabbed to death. Marcus was a brilliant and talented fashion designer and a proud member of and advocate for the LGBTQ community, but he was perhaps best known to me as the beautiful, iconic local drag queen, Maria Venus Raj. Good afternoon. My name is Christoph Kissoon, and Marcus and I are not so different. I too am 27 years old. I too am of East Indian descent. I was born in TNT, and like Marcus, I migrated for the chance of freedom and opportunity. Under the slightest change in circumstances, it could have easily been me. While it may seem like LGBTQ rights is getting better in countries like the United States, the sad reality is that in countries like mine, the very lives of LGBTQ individuals are at stake. In fact, Marcus's murder has yet to be classified as a hate crime, and there's been no justice to this date. I remember growing up in my Catholic all-boys high school, hearing slurs about batty boys and buller men, Trinidadian slang for faggot. I went clubbing and saw my generation grinding to artists like Buju Banton, whose lyrics instructed listeners to shoot batty boy dead. Apart from the decriminalization of same-sex intimacy in 2018, there are zero protections for LGBTQ individuals. So I decided to dedicate my research to investigating why, in the hopes that what happened to Marcus would never happen to another member of the LGBTQ community. I began my research by conducting a survey, and my findings suggest that LGBTQ rights remain elusive in my country for three main reasons. First, the people of TNT are very religious and perceive LGBTQ rights as a violation of their moral and religious beliefs. Second, the national government holds all the power, preventing local government from experimenting with LGBTQ rights. And third, the two main political parties intentionally maintain a culture of silence. So where are LGBTQ rights advocates supposed to go to make their voices heard? What is the average person who cares about this important issue but sees no difference between the parties or candidates to do? My research is uncovering these answers and illuminating a path forward so that one day Trinidad and Tobago can fulfill the promise in its national anthem. Here, every creed and race find an equal place.
Good job. That was quite the powerful statement. So how long have you been here in the United States?
Since 2014, so about 8
2014.
years.
Okay. And have you been back home?
Yes, I actually just came back from a vacation. I got to spend 5 weeks with my family, and it was everything I needed.
Oh, great, great. And so the research, do you have a chance to interact with people in your country as you're doing your research? Is that how you're doing it?
Yes, I've been
Okay.
collecting surveys virtually online, and I had the chance to do some field work as well when I was on the island.
Great, great. Well, thank you. Thank you. Next is our Idaho State video.
For more than 120 years, we've been teaching, moving, creating, inspiring, growing, learning, connecting, improving, collaborating, and of course, we've been roaring. At Idaho State University, we unlock potential and give our students access to an education that is focused on changing their lives. In short, our mission is simple: meet students where they're at and help them get where they want to be. Our students achieve their lifelong dreams while also discovering new goals and interests. We are a university of opportunity. We not only discover new knowledge, we also unlock doors to careers. With more than 200 program options, we successfully prepare our students for the careers of tomorrow. From hands-on associate degrees in high-demand technical fields to postgraduate and professional programs, the scope of Idaho State is truly unique. Located in the heart of southeastern Idaho, in the shadow of the mountains, our university evokes feelings of pride and strength. Our community is filled with a kinship that Bengals from around the world call home. We are a family, a Bengal family, a family of more than 100,000 proud alumni. With a 13-to-1 student-to-faculty ratio, our students have access to experts in multiple fields. Experts who are not only dedicated to their discipline, but also passionate about sharing knowledge. At Idaho State, we prioritize student achievement. From a new student's first class on campus, to walking across the stage at graduation, diploma in hand, their success is our success. As the state of Idaho's lead health sciences university, we are dedicated to improving care and quality of life. From our on-campus clinics to student placements in the community, we are well known and well respected for bettering lives around us. We are focused on building meaningful relationships that will support the well-being of our campus community and beyond. We've been changing lives for 120 years, and our roar is only getting louder. Roar, Bengals, roar.
All right, so we have 4 more to go. This is getting more difficult as the day moves on. We have some great competition here. So our next presenter is Akariti Sariki from the University of Idaho.
Each of us has over 37 trillion cells, but we started life with just one cell. That's one cell divided over and over into the complex multicellular organism that we are. Bacteria, on the other hand, start as one cell and maintain one cell with every division, so they are unicellular. When bacteria divides, it makes a clone, transferring genetic information from the mother to the daughter cell. This continues exponentially, shown in panel A, to form a colony. So a colony is made of clones or cells with the same genetic information. Based on this knowledge, we tend to make inferences about cells in a population by taking the average. But have you ever seen identical twins that were raised similarly but behave differently? Likewise, some cells in a colony can behave different from others. In my research, I use single-cell techniques like microscopy to observe cells individually. With this method, we produce the images in panels B and D of the bacteria I study called Methylarubrum extoquens. Panel B is an image of its cells from the same environment, the same population. But zoom into the part that looks like the map of Africa, where I come from, and we see the cells have different morphology. They all have the same genetic information, but behave differently. We eat food and grow. So does bacteria. For Methylarubrum, that food can be a toxin called formaldehyde. When exposed to lethal concentrations of formaldehyde that should kill all cells in the population because of toxicity, majority die, but few survive and pass on the ability to their offspring. These survivors have different genes, and I aim to understand if and how these genes enable bacteria to tolerate toxin. I do this by observing the cells in the colors red and yellow to distinguish them when the genes are active, shown in panel D. The unique ability of this bacteria to consume lethal toxins can be exploited in biotech industries by using them to get rid of toxins in the environment. More broadly, understanding how cells can be the same genetically, yet act differently, is important in the treatment of diseases. For instance, current treatment against cancer targets majority of cells in the cancer population. But studies have shown that the actual culprits are a minority of cells that are not affected by this treatment because they behave differently. Studying cells one by one can help us to understand how we can identify abnormal cells out of over trillions and target them for treatment. Thank you.
Akaridi.
Yeah.
You said you're from Africa.
Yes.
What brought you to Idaho?
What?
What brought you to Idaho?
Oh, school, grad school.
Okay,
Research, yeah.
okay. And what is your major?
Biology.
Biology. And so when I'm listening to you, you're looking for solutions that might impact both the environment and medicine. Is there one field that you're more interested in, or is it just more the study of the bacteria?
Oh, it's more the study of bacteria and basically majorly the environment, like how to use this bacteria to get rid of toxins in the environment. That's like my major focus, but like it has applications in medicine as well.
All right. Thank you so much.
Yeah.
Sorry, do I come up now?
Our next presenter is Dalton Miller from Boise State University.
All right. I'll swap you now. Thank you. All right. Chronic wounds, seen here on your left, are wounds that fail to heal for months to years from their initial onset. They affect more than 6.5 million Americans every single year, and their treatment costs northwards of $50 billion annually. Now, obviously, that's very expensive, right? That's a lot of money. But what's significantly more costly than that is the amount of pain and suffering that people with these wounds have to go through. You see, once a week, the patients will go into a wound care facility where a doctor will unwrap their wound, snag a bottle of antiseptic, and vigorously scrub out the wound site. This is to kill any bacteria or fungi present that could cause an infection, but well, as you may imagine, in scrubbing an open wound, that's a very painful medical procedure, to say the least. Now, after this, the doctor will trim any dead tissue from the wound site, rinse the wound out, wrap it back up, give the patient a pat on the back and say, well, we'll see you in a week. Hopefully that will heal. Of course, the doctor can't actually say when or even if the wound will heal. I don't think that any of you here today would describe that procedure as ideal, effective, or even particularly good, and it's to that end that we're working on a new method for treating chronic wounds. It uses an ionized gas, also called a plasma, which you can see here on your right, and it works by generating a large variety of highly reactive molecules that serve two basic functions. The first of those functions is sterilization. The molecules have the capacity to kill bacteria and fungi in the wound site, but because we're using a gas rather than the more traditional liquid, no vigorous, painful scrubbing is necessary. Now, the second of these two functions is in wound healing signaling. As an example here, if you get a cut on the back of your hand, your body will begin producing these exact same molecules to tell the cells around the wound to begin healing the wound. By introducing these molecules directly to the wound site, we can speed up the rate of wound healing simply by telling the patient's own body to heal the wound faster. Now, right now, we're testing in pig wounds just to examine safety and efficacy, but our initial results are quite promising. In as little as a 10-minute treatment, which is shorter than it often is to scrub out these wounds, we're seeing good sterilization and no damage to the pig tissue. We hope that eventually this will be used clinically, both to decrease the amount of pain associated with having a chronic wound, as well as, more importantly, to increase the rate of wound healing so that we can give these patients their lives back. Thank you. All right, I am now ready for Q&A.
I'm not that hard on you. But one question that came to my mind when you're doing the experimenting on the pigs is, have you come up with any idea of how much faster it does heal?
So we haven't yet, no. For studies like that, we're going to need to have longer examination periods, and we simply haven't been at this for long enough to get a good sense of that yet.
Right.
We've been more focused first on whether or not it's actually going to, work to sterilize anything. That was the aspect that we thought was most promising and most easily studied, and so we began with that.
So, and what's your major?
Chemistry. I don't know more broadly than that. I do a lot of biochemistry, so probably that.
And are you from Idaho? Or are you?
I am. Well, okay. Technically, I was born in St. Louis, Missouri, but I only lived there till I was 3.5, so I don't think that counts. And since then, I've lived here, so yeah.
Thank you so much. Good luck.
Thank you. Thanks, everybody.
All right. And our next presenter is Mel Anderson from Idaho State University.
My father is serving 2 life sentences for 11 counts of child abuse, including rape and torture, that culminated in my brother's murder. Critical trauma theory and literature posits that for survivors like me, trauma is both unremembered and unspoken. I disagree. I remember every moment and can speak it, and I'm not alone. After the Holocaust, psychologist David Boder recorded the oral testimony of death camp survivors. Survivors who wanted to speak were plentiful, but no one would publish Boder's research until nearly 40 years after his death because, quote, no one wants to hear atrocity. Survivors spoke. They were not heard. The very existence of Holocaust memoir, which requires both memory and voice to write, severely undercuts the belief that memory and voice do not survive trauma. Supported by tens of thousands of Holocaust narratives, my research argues for a new theoretical framework in which we shift the conversation away from survivor deficiency and towards the silence imposed by listener discomfort. The problem with trauma communication is not expressive, but receptive. Truly, no one wants to hear atrocity. Beyond the therapy circle and happy ending tropes, it's never the time or the place, and some of us don't get happy endings. It's never the time or the place to speak the very real traumas that exist in the shadow of racism, sexism, homophobia, bereavement, rape, suicide, war, or the myriad other traumas that humans face every day. This is evident when we ban books that are honest about the suffering of the Holocaust, or even when we forbid teaching U.S. history because it might, in the words of one new law, cause discomfort. Auschwitz survivor Primo Levi described a compulsion to speak after trauma, to make the rest participate. But when we teach that survivors are incapable of speech, we remove the imperative to listen, and we lose the opportunity to learn from those experiences that, while painful, are also valuable. I want the circumstances that made my survival necessary to be fixed. That can't happen while we persist in the ableist notion that survivors are too broken to participate. We can speak. My research invites others to learn to listen, even when it's uncomfortable. Thank you.
So I would say yours is one of the most personal research projects here, and I guess I'd just like to know, was there a time when your personal experience came together with a teacher or a mentor or a faculty member that inspired you to put those together?
Okay, two things. One, I served in the Army for a while, and in that function, I was able to actually go to a speech where Fanny Eisenberg, who survived Auschwitz, spoke. And at the very end of her presentation, she asked for any questions. And I'd never been in the room with someone whose story was like, I couldn't think, oh, okay, yeah, I've been there. It was like the first time. And so I asked her, after an experience like that, how do you learn to talk about the weather? And she put her hand over her heart and she said, oh, honey, you felt pain, haven't you? And she said that after that kind of trauma, you'd never learn to talk about the weather. You just find other people who also can't talk about the weather. And she talked about just being able to talk about this. And I've realized over the years that when I kind of say something about my own past, I get that, like cringe. And it made me really think. So in my first semester, when I read this theory that you can't speak trauma or remember it, I was like, oh, well, that's not true, buddies. And I've really devoted a lot of time to it since then.
Well, I appreciate it. I think what you're doing is trying to reinforce what we all need to do to be better listeners.
Absolutely.
Thank you.
Thank you, ma'am.
Okay, I believe this is our final presenter, and it is Tori Stevenson from the University of Idaho.
From the year 1900 to today, our global population has increased from 1.6 billion to nearly 8 billion people. By 2050, it's projected to near 10 billion, and the lives of every one of those people will rely on the food, fuel, and fibers grown by agricultural producers. Now, already increased demand for agricultural products has resulted in the intensification of practices that degrade the health of our soil, its ability to recover from disturbance, and to be productive both now and well into the future. Supporting this growing population is going to require identifying diverse growing practices that support rather than degrade our soils. So for this reason, my research is looking at one such practice, which may have influences on soil health in two key areas: nitrogen use and soil microbial communities. What we're doing is growing a type of crop called pulses in alternate rows with barley in a practice called intercropping. Now, pulse crops are special because they can obtain nitrogen from the air. This is a huge deal. Nitrogen is the key component in chemical fertilizer. And when we add more fertilizer to our soils, we can grow more crops per acre. But it's a delicate balancing act. Now, for one thing, fertilizer is expensive. And when we apply it all at once, it can be difficult for plants to take up the nitrogen they need before it's lost from the soil, either washed out into our waterways as pollutants or converted to harmful greenhouse gases. So adding nitrogen in excess of what our plants can take up is a waste of a resource with negative environmental consequences. Now here's where the pulse crops come in. Because they can obtain their own nitrogen, they may give us a better chance of having the right amount of it in the soil at the right time for the plants. And this has the potential to decrease our fertilizer demand and also waste. And in fact, what we're seeing with intercropping is that the nitrogen in the soil is used up more completely, meaning there's less left over at the end of the growing season to have those negative environmental consequences. So this is great news. But what we need to remember is that soil isn't dirt. It's a living thing. Soil is full of microbes, and those microbes do things like provide nutrients to plants, protect them from disease, and maintain the stability of our ecosystems. When our soils face a disturbance, like an extreme weather event due to climate change, not all of those microbes will be affected in the same way. A more diverse microbial community is going to be better able to recover from that disturbance and to keep functioning. It's healthier. And what we are seeing is that intercropping supports not only a more diverse, but also a more abundant soil microbial community than growing barley alone. So that's also great news. Now, taken together, these results suggest that intercropping may be a promising strategy for promoting our soil's health. And the impacts of this work are important not only for producers, but for those of us who rely on them every day for the food we eat. All of us.
Thank you, Tori. This is something I actually know a little bit about, so I have some specific questions.
All right.
I'm a farm girl, but my dad has, goes from crop to crop. They'll switch in the same acreage from year to year, but what you're suggesting is you do a row of lentils and then a row of wheat, and then two in the same year, all in the same crop, which is definitely different. That's fascinating. What brought you to this idea?
Well, primarily, I was fortunate enough to end up in a lab in undergrad that was looking at soil health and nitrogen cycling, and I just became fascinated with it. So when I heard that Dr. Kaler at UI was doing a project looking at nitrogen cycling, specifically with intercropping and growing different crops, I thought, that sounds like the project for me, and ended up here, and I'm having a nice time.
And your major is?
I'm actually an environmental science student, but I work primarily in the College of Agriculture and Life Sciences.
Okay, so Dean Perella, you got another one up here. Is he here still, or did he have to leave? He might have had to leave. But anyway, thank you so much.
Thank you.
All right, I think we have one more video. All right, so I am thoroughly impressed. I don't know about the rest of you, but I think the judges have a really hard job right now, and we're going to give them some time to, we've got some space for them to go deliberate and debate on the awards. I just want to say congratulations. It takes a lot of guts to get up here and do these presentations, and I know you spent hours practicing and preparing and doing your research, and I'm just super proud of everybody here today. So we're going to let the judges convene and come to some decisions. I have a few other things we need to make sure our audience does their people's choice voting. So if the judges would like to go into the room back here and gather, we'll go to the next part of this program. Okay, so first of all, we want the audience to choose their favorite among the presenters and vote for the people's choice. For those of you here, you can get on your phones. Please visit the website that you see on your screen or the QR app on your phones. You can use the QR code I suggested earlier, and it'll take you straight to the poll. It's only open for a few minutes. Please enter your vote for your favorite presenter now, and please vote only once. One year we had somebody cheat the system and we had to figure that out. But anyway, vote only once, please. And then this is the time we set aside for the audience on Zoom to ask any questions they have to any of our student presenters. Be sure to call the student out you'd like the question and to be directed to, and use the chat feature in Zoom to enter a question in the chat, and our staff member, Lauren Perkinson, will ask on your behalf. So Lauren's going to monitor that and ask any questions. Last year we kind of had a pause and then a few questions came up, but at this point in time, this is a little bit of a break. Feel free to visit, pause, and I don't know if Lauren has any questions or anything, but we're just going to take a bit of a break while the judges make their decision. Good job, everybody. So what we're going to do is announce all four of the winners, and President Scott Green will bring the award, give the award to them as they come forward. And then what we'll do afterwards is we'll regroup and have each, if you are a winner from your institution, your dean of your graduate school will come up and we'll take some photos with your community and anybody else you want in it, so we can have photo record of all the winners. So we'll regroup after we've announced and do that up here on the stage. So just stay for that if you're one of the winners. So what we're going to do is start with the People's Choice Award, and that's where everyone went online and voted from their phones. And drum roll, please, that is Allison Loyola from Idaho State University. And she's going to get $250 and a plaque here. Allison, if you get the plaque from President Green. We'll take a picture with that. Maybe we should come up here with that. Yeah, let's come up here. Come up on the stage. We didn't quite get that choreographed very well. Come on up here.
I need direction.
Yeah, you can stay up here for this when we get. Yeah, let's just line up here. All right, you can just stay up here for this. All right. Congratulations, Allison. And our third place winner today is Tori Stevenson from the University of Idaho with $500. Congratulations, Tori. Wait, let's put the. Yes, good job. Okay, and our second place winner today is Jacob Manzi from Boise State University. And it is a $750 award. And finally, our first place today, and I would like to announce Mel Anderson from Idaho State University with $1,000. I just want to say I think everyone did an amazing job. I'm sure the decision was very difficult for our judges. I was super impressed today. Go back to your campuses, encourage your colleagues to participate in this program every year. You know, it's an annual event, and the more participants we have, the better. So I thought you all did a wonderful job. I also want to again thank our judges. You're awesome. Thanks for being here. Yeah, everybody come. We'll take some pictures with your colleagues.